because in fact, the nervous system is probably the living system that most uses different forms of sensing and ways of communicating and actuators. So, sensing and actuators. I gave a lot of thought to putting my talk, which is sort of nanotechnology and neuroscience, into the context of biosensing and bioactuation, because when I thought about it, I said, you know, I could give a whole course on this in the nervous system. Everything we do all day long involves sensing. You're hearing me, you're seeing me, you're deciding whether the temperature in the room is right for you, you're deciding whether you ate too much for breakfast. All of those things involve sensing in the nervous system, and in most of those cases, you do something because you sensed it. So I look at biosensing as fundamentally embedded in the nervous system, and of course, nowadays we've taken what we know about biosensing and we're using it in different ways. So I'm going to go ahead with my prepared presentation, which is going to start out, oops, I do have an overview on my talk here. I'm going to start out talking about the brain, but I am going to go through sort of the fundamentals, and then we're going to go into talking about how you can use nanotechnology to study the nervous system and in fact study bioactuation in organisms. So, let's see here. Get familiar with the technology myself here. There we go. So, the brain here is, of course, not the largest organ in the body, but it is by far the highest user of energy. So it's only about 2% of body weight for you and I, but it consumes nearly 20%, nearly a fifth of the oxygen the body uses at rest. This is why anything that compromises oxygen to the brain is very dangerous and likely to have long-term consequences. And this is really because of the fact that the brain uses ion pumps, very ATP-using ion pumps, to maintain a differential gradient of ions across its membrane. And these ion pumps, once they go awry, then you start getting accumulations of toxic substances. The membrane surface area of the 100 billion cells in your brain is about 25,000 square meters, or the size of 4 football fields. The membrane surface, and that's critical. So membranes are what the brain is about. Membranes permit the brain to do, or the parts of the brain to do what they do. And then it's formed of cells, sproutings, that exceed over 10 to the 9th miles in branches, and every time you learn something, you sprout something new. And so one of the things the brain does for you besides biosensing and bioactuation is it changes with experience. So the fact that you were hearing my voice this morning and looking at my slides hopefully means that something has changed in your brain at the end of my lecture. So these are the learning objectives. I give a number of continuing medical education courses. In those environments, they always ask you to have learning objectives so people know where they're headed. This is what I hope to cover over the course of my talk. I'd like you to learn the unusual structural and functional features of brain cells, and they go together. I'd like you to know the bases of biosensing and bioactuation by neurons, which are the primary cells that cause biosensing and bioactuation in the brain. I'd like you to understand why new technologies are necessary in neurobiology. And then I'd like you to recognize the importance of materials, the 3D environment, and novel imaging to learn about brain cell function. And at the very end, I'd like to give you some examples to stimulate your thinking about questions to answer and novel discovery applications. So then, I'm going to spend quite a bit of time talking about the unusual structural and functional features of brain cells. So the brain is formed, actually, it all starts with the fertilized egg. And I like to bring this up because it's quite remarkable that we all start from a single egg, and then you get the kind of cells that I'm going to be telling you about, which are really highly unusual. So here is once you get to the point where you have the cell types going into the various layers of the body during development, you get the primordial cells that are going to form the nervous system. These are called neuroblasts, and here's one that's divided in two. Those neuroblasts, toward the end of their developmental sequence, form two cell types, the neuron and the glial cells. The neural cells are the cells that are responsible for sensing, transmission of information, integrating experience, and then feeding out through neurotransmitter systems to actuators, which may be muscles or glands or blood vessels. The glial cells, which are named from the Greek word for glue, are the cells between the neurons. And these are the cells that have been thought to be support cells. They actually play a critical role in the functioning of the nervous system. My purpose this morning is not to focus too much on the glial cells, that's the story in and of itself, but to know that they come from the last cell division, or the second to last cell division, just before a particular brain structure is formed. So we have about 100 billion neurons. I don't know how they got this up, but that's what the calculation is. And then with respect to the ratio of glia to neurons, this is actually in dispute these days. Even in my lab, we've been discussing the new evidence that previous reports would say there are 50 to 10 glia for every neuron, but it's emerging that that number may not be right, and it may be more like 1 to 1 or 1 to 2. Nevertheless, there are glia and there are neurons, and they came from the same precursor cells. The neurons look something like this if you stain them for, this is in fact stained with a dye that's going out into the membrane. And what you can see here is the cell body, which is equivalent to this area in the neuroblast. It's where the nucleus resides, where the energy, most of the metabolic enzymes, the Golgi. I know Scott Scheiton told you all about these in the first day, so I'm just kind of reminding you. All the basically business housekeeping of the cell is done in the soma or cell body. But in neurons, the cells go through a developmental sequence whereby they extend these very long processes, which become highly branched and highly elaborated. And this is just to compare these with the glial cells, which have various forms. I have two here, but they also have processes. They're somewhat different in shape. They're certainly not as long and not as elaborated as they are in the neuron, but they are, again, more like neurons than they are like other types of cells, like fibroblasts or muscle cells, other kinds of cells that you might have experienced. Now, I think it's useful to think about the process of neural differentiation, how you get from one of these early, relatively undifferentiated, nearly spherical cells to something that's very highly branched. And this is from this is relatively early paper now, but many studies were done over 100 years ago. Your teaching assistant, Larry Millett, was a graduate student in my lab, and he's preparing a paper from his thesis on 100 years of neuron culture. So neurons have been studied for, this is the 100th anniversary, the centenary of when we started studying neurons and culture. And so that's when we started looking at this process and observing it very carefully. But nevertheless, in the earliest stages, if you put these cells in culture, they extend ruffling membranes called lamellipodia, and then within a few hours they start to extend minor processes or protrusions from the cell. And at this point, these protrusions are simply called neurites. They're difficult to tell one from the other, but they're characteristic of neurons and not of other cell types. And then, as another day goes by, we see that a decision is made. A decision is made that one of these processes will become different than the others. And that particular process becomes very elongate. This is the leading edge down here. The name of this process is the axon, called axonal outgrowth. And the axon becomes the transmission end of the neuron. Neurons are polarized structurally and functionally, and the axon is where the information flows from the neuron to whatever is downstream from it in the communications pathway. The other processes then continue to grow and differentiate, and oops, I put a little animation in there. So there's the neuron outgrowth getting longer and longer and longer over the days it's observed. But then we have the dendritic outgrowth, which then becomes elaborated over several days. And part of that involves changes within the structure of the dendrites, which you can see as these little, they're called buttons or batons many times, these little enlargements, and I'm going to be talking more about those. But this is just an overview of what the differentiation of a neuron is like. There we go. Oh, I do. So the wiring of the nervous system, and that is how do you go from a cell to a brain? And that involves a very intricate process whereby the single cells connect into very complex circuits and networks. And what's important here, I think we fail to appreciate this many times, is that this is a self-organizing process. You don't have to have experience in the world for your brain to grow, to form connections, to form functional circuits, to reach out into the body, make your toes wiggle and your heart beat with a certain rate. All of these come from self-organization, and they're the result of directional growth of the axon to the target. And what we have here, labeled with rhodamine phalloidin, which labels filamentous actin, is a very highly undulating tip of a growing axon. And what that axon is doing is it's searching around the environment and sending out very, very fine processes, which are actually hard to see in this movie. But those phylopoid, those fingers, are testing the environment chemically, can be also be testing it physically and possibly mechanically. But they are then looking to see where this action should go. So that's the exploratory, out into the big wide world of the brain and body part of the neuron. Wiring of the nervous system, self-organization, also relies on dendritic development. And dendritic development has been much less studied than has axonal development. In fact, there's a huge literature. I didn't go to look how many papers there are, but if people studying how the brain wires are all looking at the axon. There's much less done on those dendrites, those other extensions from the cell. And let me just say here that if the axon is the output side of the cell, the dendrites serve as the antennae. They're the input side. So they have many fine processes. They establish connections that are going to be bringing the information in to this neuron. And early in the process, within this image, actually Larry just came in, so I should say bye Larry, and this hippocampal cell, which is from a part of the brain that's involved in memory acquisition and, oops, what happened? Oh. Let me just look at that as perhaps I didn't touch the screen. Oops. Now we don't even, no. Okay, preview. So, at 10 days, let's see, this is an immature hippocampal neuron at 10 days in vitro, and what you can see here is, with a dye that's labeling the cell membrane, not only the form of the major processes coming out of the cell body, but on it you can see these very, very fine, looking like hairs, but not hairs at all, very fine extensions called phylopodia, or basically they're like finger feet that are going out and exploring the world around them. And then, so this is a confocal image of the phylopodia. Now I'm going to show you some images that were done by Larry, and this project's being continued by Raj Iyer in my lab. And that is looking at the behavior of phylopodia in real-time, non-invasive imaging with a new method developed in the Popescu lab, scattered light interference microscopy. And this is simply using the refractive differences of the constituents of the cell to bend the light so that you can see structures in the cell without any dyes or any other kind of interventions. And what you see here are two neurons lying next to each other, and these are going to be hippocampal neurons again, primary hippocampal neurons from lat. And these are the cell bodies, and what you see here with these differences in refractive index are where the nucleus is lying. And then here is a meshwork of processes extending from those cells. In the next slide, we have what I hope is a movie. There we go. And so what you can see now is this is, I don't actually have what the time lapse on this is, but what you can see here are the phylopodia waving around, searching around. You can see how active they are. This is a flattened membrane like a lamellipodium that's flattened out on the substrate. But also, you can see the migration of materials through the processes. Some go one way, some go back, some of the same ones go both ways. But what you can see is this is a very dynamic situation. These cells are not just sitting there and sort of slowly moving things along. There's constant motion. There's constant activity where the cells are moving things, transporting materials, searching the environment with these phylopodia. And some of this will stabilize as the cells mature, because the phylopodia are only present transiently during the early stages of development. And this is showing you something that I wanted to make sure I emphasized, and that is not only do you have the phylopodia coming out of this tunnel, but you have the elaboration of the branching of the processes that are extended from the cell body. And this is an analysis of the primary, secondary, and tertiary branching that Larry did over at the visualization lab at the Beckman. And you can already see, does anybody see the axon? And what you can tell about the axon is already it's looking very different from the dendrites. It has some branching, which isn't apparent here, but much, much, much less than the dendrites do. Very many fewer phylopodia coming out, and it's actually somewhat thinner, and it's going out very, very far away. In fact, in the case of some of the spinal motor neurons, the longest spinal motor neurons are actually going from the region of the sciatic nerve in the lower spine of the elephant down to the elephant's toes, bottom of the elephant's foot. So the axons have the ability to become extremely long. And this, of course, is an interesting question. How can you make a protrusion on a single cell become so very long? What does it take to support that? And you can ask Dr. Saif that, who's giving the next lecture. That's, I think, an area he might have answers to. So then, the next stage in the development of the dendrite is spine formation. And spine formation is something we know happens because that's what you see in the adult cells and in the brain. This is a mature hippocampal neuron after 33 days in vitro, and it's stained with antibodies, which is what we call immunocytochemistry. So we have antibodies against known, in this case, proteins. One of them is microtubule-associated protein 2, which is in dendrites, and so you can see the blue here is labeling the dendrites. And we have green, which is labeling synapsin. Synapsin is a synaptic protein. When synapsin is expressed, it's localized to the dendrites. It suggests that the dendrite, it's localized to the synapse. It suggests that the synapse may actually be active. The synapse, if there's a way to keep this from searching so fast. Anyway, does not tell you the synapse is active, and I actually haven't told you what a synapse is. So the synapse is the point at which two cells in the nervous system communicate. So as the axon grows out, it's going to form a synapse with a follower cell in the circuit, or with a follower cell, which is an effector cell, like a muscle, or an endocrine, a gland. But here, the fact that you have all of these green spots, which are actually sitting on the surface of the protrusion, suggests that this is a highly innervated neuron. And then we have red, which is stating filamentous actin. in various places, and some of that's obscured by some of the other brain staining. So spines, and then here I have it back up here. Spines and the staining with synapsin indicates that this is an active neuron. Now, I have superimposed on this. Be patient with this. Oh, I know what I did. Yes, information flow. So I decided it would be useful to emphasize the flow of information in this cell. So we have very fine branches here. When a synaptic input activates this process, the information, if it's coming in on two of these, could be additive. We see changes in the activity based upon ionic fluxes that are coming into these distal dendrites, and depending on the strength of the excitation, the information will flow along the membrane of this dendrite. We may have nearly simultaneous input from other parts of the cell, and these may converge, go into the cell body. You may get input from other cells as well. And then I made this the axon. Then the information will be integrated, summate, and flow out of the axon and down to the next cell. So that is basically the nature of information processing on the cellular level within the context of these extensions. Now, that is not the next thing.
So what type of form is the information?
Excuse me?
What type of form is the information?
This is electrical. All of this information at this stage is electrical. The rapid flow along the membranes is electrical. And nevertheless, I had another image of some spines, but we can move ahead. So I wanted to make the point that neurons and glia are not separate entities. I did say that they were supportive, and I indicated that the glia, that they come from the same precursors, but in fact they intertwine. They interact spatially, temporally, and functionally. So this is in culture, but even in culture you can see that there's a close physical relationship, spatial relationship between the neurons, which are in green, and then the glia, which are in red, stained in red. And these are simply stained with antibodies to structures that are specific, usually cytoskeletal structures that are specific for those two cell types. It is the case that in culture, neurons grow much more happily if there are glia included in the culture. Either the other speakers talk faster than I am, or it's very rare that someone would tell me that someone else talked faster than I did, but nevertheless, we're going to just persevere with this. But one of the things I wanted to emphasize is if this is the relationship, the physical relationship of the neurons and the glia in culture, this is the physical relationship in the brain. And so what we see here is that there are intricate circuit and synaptic architectures. This is in the mouse cerebral cortex, which is the part of the brain that's overlying basically right under your skull. And it's larger in animals that have more cognitive abilities and more capabilities of doing things with their brain. So it's an interesting part of the brain. Thought to be where some memories are stored. But nevertheless, these are stereo reconstructions from 134 ultra-thin sections that then were stained for something that was expressed in one of the layers of the cortex. So these are these are cell bodies of neurons in this cortical layer. These are basically what we call pyramidal cells. But what you can see here is the meshwork of interactions that the processes are forming. And you can see that the processes are studded with these little dots, which are the spines that have been formed on the dendrites, and actually are the sites at which synapses are formed. Now, what about the glia wrong way, in the living organism? And I took this from a Nature Neuroscience cover because I thought it was so spectacular. And it was focusing on the role of glia in disease. But what is remarkable here is that these glia were stained not with a cytoskeletal marker, which is what all the previous images I showed you were. And in fact, most of the staining of glia, as it is with neuron, if you're just looking at the general shape of them, is done with antibodies that recognize the structural proteins of the skeleton of the cell, the cytoskeleton. But in fact, this is using a membrane dye. And with a membrane dye, you see something completely different in glia. I heard a talk on this last winter at the Winter Conference for Brain Research. And indeed, one of the researchers there affirmed that this highly elaborate invagination is characteristic of glial membranes, completely different than what you see in neurons. In neurons, the shape of the neuron is very much like what you see in the cytoskeleton, but not in the glia. We don't know what this means. And the other interesting relationship between neurons and glia is that whereas neurons are sending their axons very, very far away and interacting with other neurons or follower cells, and dendrites are way out there, all the places that neuron's getting information in. Glia have very distinct local domains. They're distinct from neurons in the way they interact spatially with their environment. We don't know why this is. We know very, very little about glia, as a matter of fact. So glia is one of the big new areas, but here, glia regulate brain vasculature, therefore they're important in ischemia, which is oxygen deprivation and stroke, and myelination and repair. So they basically we're quite sure they have a significant role in neurodegenerative diseases. So, oh, and these are human. They're from another cortical region. So I guess this should have been used for the other one, but this is simply showing you the three-dimensional arrangement thing, how the pyramidal cells and their processes with the boutons. And then these are, the blue is marking nuclei. This would be glial cells or other types of cells that are interspersed with there. And then the red dots are the spines or boutons of other cells that are coming into this region and being active there. So now I'd like to talk about, I could give a lot of lectures on what I'm going to show you in the next two slides, but I decided to restrain myself and simply tell you an overview of how neurons are specialized to sense, integrate, and transmit information. Sense, integrate, and transmit information, and to change with experience. And so what we have here are the most common kinds of schematics that you will see in textbooks on the nervous system. This is from Scientific American, which is, like a textbook. And basically, these are looking like those pyramidal cells I just showed you. This is the schematic. What I showed you is the reality. But this permits us to then look a little more carefully at how neurons are organized in beginning elements of circuits and then how they work functionally. So here we have one neuron, here's this nucleus sitting in the soma or cell body. We have the dendrites coming in. Here's the axon. And then here is the site at which, various sites at which this axon is innervating follower cells. This is where the synapses are formed. So then this is the dendrite of these other two cells, which is receiving information, in this case from several processes of the presynaptic cell. The number of synapses that any single cell makes in the brain is been estimated in the tens to hundreds of thousands. It's tremendous. And this is why making computational models of the brain is difficult. I will leave it there, because that's not what I do. But people are doing it, and with some success, because not all the synapses are equally important, but certainly if they're maintained, they're doing something as well. So the way that information flows is based upon electrochemical gradients across the lipid bilayer of the membrane. And if you really want to know about this, what I'm telling you is not going to answer your questions. But basically, this is, for instance, looking at the lipid bilayer of this part of the axon. There is an ionic disequilibrium across the membrane of the whole neuron, such that sodium is high on the outside, and the basic way to remember this is it's as if the neuron were living in a salty sea, because that's where it started out. And then potassium is high on the inside, and neurons work very, very hard to keep calcium low inside. And so this membrane has a certain rest exchange, but there are all those pumps that were using all the energy in the brain, they're keeping this gradient different. When a signal comes in, it can either directly by a chemical signal binding to an ion channel and changing its three-dimensional conformation, or by binding to a channel which causes a voltage change, a change in the charge distribution across the membrane. That can cause a change in the conformation of that channel, or by an indirect method which causes the receptor to make smaller molecules inside the cell which come back and then affect memory. So you can tell this is a complicated process. But there's many ways to regulate the activity. But what happens at a certain point is that you get enough change to keep the signal, the electrical change, propagating down the membrane of the dendrites, so that when it comes to the axon, this is probably good for memory, the fact that it keeps coming off, right? So when it gets to the axon, you have a very high concentration of channels that are basically, regenerative, so that they basically self-perpetuate the excitation. The regenerative capacity is similar to that with a flush toilet, whereas once you reach a certain point, the information will flow down the axon without any more input of any kind. And that this permits the signal to rapidly transit in an all-in-one fashion down to the next cell and communicate across the synapse. And here we have a schematic of the synapse. The synapse has two components. One is called the presynaptic terminal. And I've already told you enough about the brain that you should be able to tell me where the presynaptic terminal is on the cell. What? It's at the end of the axon. So, because that, remember, is where the information is flowing down, and then that's where the cell sends information out to the follower cells. So you have this specialization, presynaptic terminal, where we have a specialization of the cell that's ready to release neurochemicals when the signal comes. So when that signal comes down the axon to the presynaptic terminal, it mobilizes the things that are stored in vesicles there, and they're released into the space between the cells. The cells are actually physically separated, but it's not a very long space. And so by diffusion, the chemical neurotransmitters, they're transmitting information from one neuron to the next, or the follower cell. They diffuse across and bind to receptors on the postsynaptic dendrite. So that's how the information flows from cell, axon, synapse, dendrite. Most of the communication in cells that's rapid, in brains that's rapid, takes place by neurochemical transmission of some kind. Neurochemical transmission in and of itself is extraordinarily complicated and uses a diversity of chemical messengers. These messengers can be elements of intermediary metabolism, and I'm sure you've heard of them in the popular literature, like serotonin and dopamine. They're chemicals that things go awry with some of the major neural diseases, especially neurodepression, mental illness, and so on. But there's a whole other class of chemicals, which are neuropeptides. Those neuropeptides are also communicating and modulating activity, and a single synapse can have multiple kinds of messengers. And so information flow is very complex here indeed and has lots of ways of being controlled. Now let's see if we can go over here to what I prepared. So neurons are functionally polar. At the cellular level, the information flows from the postsynapse, the spine, the dendrite, the soma, the axon, and the presynaptic terminal. At the circuit level, it goes from cell to cell, it goes throughout networks, and it goes to systems. In this case, for instance, it goes from the brain to the autonomic nervous system to your heart, where it regulates heartbeat. So this is basically a polarity in not only the structure, but the way in which information flows. And the other universal that I could say, now that I've said that, is that although information flows primarily in one direction, it doesn't only flow in one direction. And there's almost always some way of the brain and the neurons sensing what happened with the change it made and whether it was the right change to make. The other point I'd like to make here is that these changes are dynamic. The electrical changes take place at the millisecond level. But you have changes from millisecond, second, minute, hour. You have changes during development and over the lifespan. You have changes with experience. And the experiential changes take place over these sorts of time courses as well. So they're endogenous changes and they're things based upon what you're doing, and they take place every day and every year. So, a lot going on in your brain. Now let's see, oh yes, so integration across levels of analysis and disciplines is crucial to understand how the brain works. And the other things that I'd like to emphasize, which we touched on, is that the language of the brain is multiple, the brain speaks in multiple languages, I'll just say it that way. Primarily, this fast transmission is electrical, and it's based upon this potential energy differences due to the differential ionic distribution across the cell membrane. Then it's also chemical, mechanical, and electrical at the synapses. So you can have electrical synapses where information flows electrically between the two cells. And then you can have the physical forces of the mechanical interactions also affecting communication. So, yes, and that I've shown you examples of model neurons. And these are the neurons, all of the things that we did our in vitro work with were from the hippocampus, because those are very, very interesting cells, and we know a lot about them from the animal. But there are many diverse types of cells in the nervous system. There are sensory neurons, which are specialized only to sense different kinds of physical and chemical inputs. What are some of the sensory neurons that you guys already know about? They're the photoreceptors in your eyes. For instance, there's your taste buds. There's the sensation of pressure. There's pain. Auditory. Dr. Fang is going to come next week and talk to you a lot about auditory sensory neurons. And so listen to him carefully when he comes in and gives that lecture, because he works in this area. Let's see here. There we go. Then we have interneurons. And interneurons are like these neurons. They're neurons that communicate between two neurons. Most of the cells in the brain and body are interneurons. They work between neurons. And they have many, many, many important roles. They have roles of integrating function, in organizing function, in encoding changes in the nervous system. Decision making takes place with interneurons. And then we have specialized cells, which are motor neurons. And these are the ones like in the elephant's leg and in your leg and my leg. These are the specialized neurons that go from the nervous system to actuators, to effectors in the body, like muscles and gland cells. They may innervate the muscles, the specialized muscles around endothelial cells in the blood vessels. So those are basically on the output side of the neuron, of the nervous system. And then here we have the glial cells, which I want you to keep in mind, but probably the last time I'll say anything about glial this morning. So, oops. So basically, the goal of neuroscience is to understand the brain from the level of synapse to thought. Now just think about that. synapse to thought. I've shown you where the synapse is. The synapse is this very, very fine structure on a single cell in the brain. And thought is something that I hope you're doing now that's involving a large part of your brain, you know, from some of the imaging work that's been done on the brain that involves large parts of the brain working in synchrony or at least in concert. So neuroscientists, and this may include some of you in the future, have a large task before them. Plenty of interesting questions to answer about the brain. And the other thing we notice is that the more we know, the more there is to know. So to understand brain functions, we need new kinds of measurements. And I'm going to tell you about some that primarily we're doing in my lab, but not only in my lab. And these involve using microfluidics to design microenvironments to better understand what's happening at the level of the single cell, small groups of cells, or in some new work we're going to be doing in large groups of cells. We're doing some work on proteomics, and that's identifying the players here that are released from specific types of cells. So we have a whole catalog of the parts list of potential neurocommunicators. We have non-invasive physiology, which is really the wave of the present in the nervous system, and that is using genes, molecular biology and genetics to put reporters in so you can look. That's a kind of minor invasion, but once the cell is in there and living, you can use it to observe in the brain what the cell is doing. My goodness, I don't quite understand why I'm having so much trouble with this today. Nevertheless, we will persevere. So now I'm going to be moving into talking about methods to study the brain. So I hope I've convinced you that the brain is a complex and dynamic network and has this basically complex network of cells that changes over time. And these depend upon the microscale cellular and molecular scaffolds and gradients. And so these basically differences in the space and the time environments of cells cannot be accurately addressed with the kind of neural cell culture methods that are based upon seeding cells over a homogeneous substrate and bathing them with a homogeneous solution. Are you homogeneous? No living things are. That's a way in which we first approached studying complex systems. But now we are at a point where we can control the environment and control both the space and time dimensions of what the cells are experiencing. And this is simply to show you, I think there's a wonderful video here from Albert Fulch's lab on ways of using microfluidics to control environments. But basically, and I don't know how much is coming for me on microfluidics, but I'm just going to point out that this is a science and technology area that's using very, very small amounts of fluids and channels with dimensions of 10 to hundreds of micrometers. And so this is basically a channel that was developed. Larry, did you develop this one? Yeah, for culturing cells with flow in these microenvironments. And so these provide new opportunities for controlling space, time, and the environment, chemical environment. And then they also let you control things around individual cells, not just cells, small populations or clusters, but individual cells that let you measure and deliver chemical and physical cues. This is something we haven't actually been able to do previously, is to really very acutely and with great resolution control the environment. So this gives you discrete access to basically networks, neurons and glia, and their subcellular processing departments. So basically, this is an area that's growing, and I actually had to update it this year. I had to go in PubMed and look for terms microfluidic and neurons. And this is basically the rate, there's a rate of growth with these two separately joined in this way. You get the numbers with different searches. But what we already have in the first 6.5 months of this year is it's equivalent to what we had in all of last year. And may I say, my lab has contributed to this. And so I'm expecting there to be a big increase, and so next year we'll have to see where we were at the end of 2010. And the fact that the Society for Neuroscience meeting is now having sessions where this kind of work is presented, they're still one of the smallest sessions there, means that this is coming more into mainline neuroscience now. And some of the applications of microfluidics and neurobiology include uses in gene profiling, delivery of genes, and peptidomics. It includes drug effects on neurotransmitters, stem cell differentiation, taking stem cells and putting them basically in a controlled environment and seeing if you can control how they differentiate. And then also in nematode biology, where people put these little, very small roundworms into the micro device and look at their behavior. And there you have a whole organized body, the roundworm, and you can basically look at its stimulus response characteristics. And then for compartmentalizing cells and cultures, controlling network connectivity, and then for studies of spinal cord and neuron repair. And this is an area that really has a lot of promise, where you can look at what's happening when the axon gets injured, and then when it tries to repair itself. Dendritic filopodia in spines and axon guidance. So these are really a large number of applications. Some of these areas are quite mature, for instance, in axon guidance. And some of them are just sort of spontaneous. They come and they go, like nematode biology. And I expect to see this used very widely in stem cell differentiation and drug effects and peptidomics and so on, when you can really get to the level of analyzing what's happening at the single subcellular points. So, having gone over the background, I'd now like to talk about some specific experiments involved in materials, 3D environment. I've already talked a little bit about novel imaging to learn about brain cell function. So, micro devices and neurons. Things that need to be considered are many. Neurons are the most sensitive cells to culture. We're asked all the time if we will help people develop primary neuron cultures. Very few labs who aren't in neuroscience do that. It's not that you can't learn, but you have to sort of think like a neuron. You know, what is it that's changed when I take this cell out of the body that's not permitting it to survive? I've already told you that the metabolic rate of neurons is very high, so they're very susceptible to anoxia, low oxygen tensions, or lack of oxygen. They're also sensitive to substrate texture, substrate chemistry, components in the medium, growth factors or the lack of them, temperature changes, level of calcium. Remember that I said cells go to great lengths to keep calcium low inside the cell unless the calcium has a specific function. And if calcium goes high or becomes unregulated, that cell is slated to die. So really take very good note of what's happening with respect to the calcium. And other considerations are the fabrication materials, whether you're using silicone, glass, quartz, or silicate, or polydimethyl-sulfoxane, PDMS. I think you guys are using this in the labs here. And most of what I'm going to be talking about has to do with PDMS devices. It has advantages in that it's thermally stable, biocompatible, low cost. You can scale it up or down. It's transparent, gas permeable, and it's easy to fabricate by soft lithography, as I think you're discovering. It's not the only material, but it's one that has a number of advantages. It's probably not the material you would use in a device that you were hoping to implant in the body. And when I call the National Institutes of Health about opportunities for funding in neurotechnology, that's the first question they ask. Are you working with a device that can go in the body? This is not it, but there are some that can't. But that's moving forward, you have to think about this as you ask different questions. A device in the body is either for sensing or treatment or maintenance. A device outside the body is for studying, perturbing, controlling, and learning things about the cells you couldn't learn before. Yes?
What's the reason? Well, what is the reason why?
It's because it depends upon what the mandate of that Institute at National Institute of Health is.
So it's just biocompatibility?
You mean, well, this one wouldn't go in the body?
Yeah, why?
Because it's rigid. I would say it's rigid. It involves flow of substances. If it's going to go in the body, I would think that you would want it. Yeah. If it's going to go in the body, it would seem to me you would want it soft. You know, our organs, unless they're holding things up, unless they're part of the skeleton, are soft and pliable and flexible. And you would want it to be probably vascularized, because that's what brings nutrients. So there are a lot of issues. I don't know that there are any devices that, unless they're basically, I think people are putting in, well, let's think of what they are. Heart pumps, stents in your blood vessels. They're interested in spinal repair. But none of those are really truly biocompatible. And even like with the stents, you know, most of the stents don't interact with the flow of blood in the same way as blood vessel, and so they don't, you know, they're not really as good. That doesn't mean you can't put them in. They're better than the alternative, which is dying soon after you get a rupture there. But I would say that these are, this is my guess, that these are probably not the components that are going in the body, but prove me wrong, you guys, because to me that is a, you know, these are great. We have found them to be, we have found them to be very, very useful for stents, because they have a lot of positives. So, now, I brought this up just to remind you, before I go on to the next things we have to consider, that something that's very important to have been established, and that is that neural differentiation in vitro, in the glass or PDMS dish, that this came from the early experiments where everything was in a glass chamber, recapitulates ontogeny. Ontogeny is the development of the organism in nature. And so these changes in differentiation of the neuron are the very same ones that happen in the brain and body. So this is good. It gives you confidence that we're using the right, you know, that we were able to get conditions that are in many ways mimicking what's happening in the body. So then going back into more considerations here, and our ability to design microenvironments that probe neurons in new ways, we have to think about what is it that we can ask with these environments. How would you design your experiment to take advantage of what these fabricated environments can tell you? And these are some of the considerations that we have and others have explored. And that one is, how can 3D microstructures affect neuron behavior? Another one is, how can the physical environment shape the development of the neuron? How can the physical shape or, what the physical conditions that the environment offers determine or in some way modify the way the neuron develops? And how does the chemical environment shape the development? Then related to this is, somebody should have this fixed for this. There. What chemical signals are released locally at different synapses? That's one of the questions. That's what actually got my group first involved. We don't have the answer to this, but this is what we started, the reason we started to do microfluidics. How can we really determine what's happening in a single process and what it means? And then how mechanical signals shape neuron development. The last point I am not going to talk about. I'm setting this up for my good friend Tahir Saeed, who is the next speaker this morning. And so you can ask him any questions you want about mechanical signals, because he is an expert on that. But we're going to be talking a little bit about these other points, and let's see what we have next. So, microstructures and neuron cultures. So, you can have groove structures. These are different kinds of microstructures that you can present the neuron with, that you can fabricate. Groove structures, nano-sized columns, different arrays, regular or irregular arrays of millimeter-sized columns. And why might they be interesting? Why would you want to stick these things in the environment? Well, it's possible they could provide more biocompatible surfaces They could be more like the environment the neuron normally sees in the brain. Remember, the neurons are very highly intricate and intricate meshwork. They're not even, I don't think a net does them justice. And then they can be platforms for studying responses and platforms for manipulating cells. So here we have just some gee whiz observations that came from a paper that originated from Ralph Nuzzo's lab that we interacted with Jen Hansen-Shepard on. And this is what she constructed, are these columns. Then she seeded hippocampal neurons on the column, on the platform here, on the PDMS. And what she found is that the axons tended to grow toward certain spacings of columns and then to wrap themselves around them. And so they really liked having the structure in the environment. They were somehow very positively attracted to this. This is an otherwise neutral environment. This is an environment without chemical signals, without trophic cues, directional cues. The cue is this physical, three-dimensional microstructure in the environment. And it's really quite remarkable the way the red here is the axons coming, winding around. And I think this is her drawings or histograms of how different spacings, this is clearly wrapping around this column and this column and this column. And then when the spacing was much closer, we saw much less wrapping. So from this, you can deduce things about the kind of microenvironmental, basically, topography that the processes are interested in interacting with. Now, here is another, this is not from my group, this is from Gallo's group, published in Biomaterials a couple years ago, but I like it because it shows that axons navigate around corners. And this is basically a TDMS environment with these little channels here. And here's a cell, a neuron. And what you can see here is that this axon has gone up here and gone around this corner, up here and gone around this corner. This is basically without other kinds of cues. So there's something about the shape of this change that the neuron is able to recognize. It's not going down the middle, notice. It's going along the edge. And I'm going to show you some other pictures of, here's going around these corners too. So basically, axon guidance and extension are regulated by three-dimensional constraints. So that's just one image. Now, I think I have, oh yes, this is a paper that came from Ann Taylor's thesis work with Carl Kottman, who's a neuroscientist at Irvine, and June, who is now in Korea. He was at that Irvine. It's a very nice Nature Methods paper. And basically, they have two chambers here. They seed the cells on this side, and then they have interconnects between the two chambers. And when you look then at a cross-section of the chambers, what you can see here is a very, very fine interconnect between the ports on these two channels. And let's just see what comes next. Yeah, so she used Texas red, which is simply a dye, to show that this is a fluidically isolated compartment. Otherwise, you see Texas red going through these little interconnects. You see these little green things here? Those are the cell bodies of the axons that she's plated out on this side. And then here, there are these grooved interconnects. And what you can see here is the growth of the axons down these very fine compartments. And they actually labeled this side of the chamber with S35 methionine, which is simply a radioactive sulfur on this amino acid. And it just tells you where that is incorporated into proteins on this side, and it has to be done by cells. And so you get an idea of where the cell body is and where this process came from. It's really a control. And basically, it's showing that the axons are basically attracted to growing down these interconnects. And so the interpretation is that when there's flow, the hydrostatic pressure helps polarize the neurons. Now, what I'd like to say is she's got another paper that came out in Neuron about 2 months ago, very recently. And it's on using this method in neuroscience to look at plating cells on this side and this side of the device, and then looking at synapses formed within the interconnects. And she's actually looking at the expression of synapsin, that synaptic marker protein. And then she's looking at RNAs, ribonuclear proteins, that are transported down the synapse. And this is a very interesting area in neuroscience, the idea that RNAs go to the synapse where they have specific functions in what the synapse does, which is respond to experience. So that, I would encourage, if you're interested in this area at all, go and look for her new paper in Neuron. It's a neurotechniques paper in Neuron. Now, the next thing I want to do is talk about neurochemical cues. And basically, this is a very well-established area, that there's a range of axonal guidance cues and modes of interactions. And these include molecular cues that are attractive, which is what's happening here, where we have, these are the axons, and they're basically being attracted to, these are other axons or cells, or they're repulsed. That is, there's something about that chemical cue that causes the process to turn away from what could be innervated. So you have positive cues, which will attract or enhance growth toward a target, or you have repulsive cues that move it away. And you can have, these are basically cell-based. You can also have diffusive cues, which are attractive or repulsive. And so we know something about this, and this is simply a schematic of this. So we have the contact cues and diffusive cues. Now, amongst the known molecules influencing axon guidance and action are many of the cell surface, the extracellular matrix molecules, such as the tenasins, and then that's on the repulsive side, and on the attractive side, we have other cues, and among them are laminins. I'm going to be saying a little bit about laminins. We also have transmembrane proteins like the semaphorins, which are in the membrane and can bind to, on the extracellular side, to extracellular signals or substrates. And on the internal side, they then activate the synthesis of signaling molecules that cause intracellular changes. This is, this is complex business here, how this happens. But we, those of us sort of in the trenches, and by that I mean as students, can use the information that's come out of this kind of work to look at potential candidates for then studying in these very, very refined microenvironments to see how they affect the wiring or the behavior of the neuron. Now, here in a cover that I get to, again, give Larry credit for, June, it's about 6 weeks ago in Lab on a Chip, is a scanning EM he made of a hippocampal neuron. I bring this up again because this is the real thing. This is what, in the scanning EM level, the dendrites look like the soma, and then here's the axon. Notice all the branches here. But in this study, he developed substrate cues to look at in a polarized substrates, a polarized environment, and just to use the laying down of the substrate as a way of understanding how you can guide the behavior of the processes. This is showing that in basically a flat, undifferentiated environment, unpatterned substrate, that you get a random meshwork of neurons. And so here is a scattering of neurons. The processes are sticking out every which way. These are hippocampal neurons at 7 days on the uniform poly-D lysine on the glass. And the red are the axons because of their cytoskeletal protein, and the green the dendrites. And what you can see basically is that this is truly like a meshwork, and there is no pattern. Now compare this with the topography of the cells on a pattern substrate. I apologize because I know this is a complicated slide, but basically the way in which this was done is to lay down a laminin. Well, this is basically a 3-chamber microfluidic device with interconnects between each of the 2 outer chambers within it. And basically this is laminin that's been laid down on this chamber, then it's washed off, and then polyol lysine is laid down over everything. And then it's rinsed, and then the neurons are put on. And basically, the neurons are in this zone here. And what's interesting about this is how do the neurons respond to the difference between a substrate which has a laminin, that extracellular attractive substrate cube, and one that doesn't. And what you can immediately see if you go down here is that each one is a different neuron. And what you can see is they're almost all heading west. All the axons are heading west. These were traced by camera lucida. And this is simply showing you the differences in the directionality of the axon navigation in the presence of the choice to go on the laminin substrate. And you can see that it's very highly significant migration toward the laminin. So that's evidence that there can be a pattern substrate that gives you information that would then align the axons in distinction from what I showed you in the last slide about the random distribution. And this is even more compelling. In fact, one of my colleagues, when he saw this, I sent him a paper, saw this, that is extraordinarily compelling evidence that if the soma migrates, the axon protruding from the soma migrates toward the laminin, and then happens to go in another direction, so it's coming back in the gradient zone, it turns and goes back to the laminin zone. So it really prefers laminin, and that's undoubtedly a laminin concentration that's permissive for what that neuron wants, what that axon wants to do. So they can detect substrate gradients, and they like substrate gradients. Now, I'm looking at the clock. I have 5 minutes, right?
That's correct.
Okay. So what I'm going to do is, this happened to me last year too. I don't know why I thought I didn't have even minutes to talk about. So basically, I'm going to, it's okay, because this paper is published. So this is another paper where we're talking about low density cultures. So now frame shift. I just was showing you about culturing neurons in relatively larger densities, but densities where we can still look at where the axon's growing, to some experiments culturing neurons at extremely low densities. And this is kind of pioneering work because there are no glial substrates, glial cues, and there are no trophic factors. And this is where we're trying to get an environment where we can actually discriminate what peptides and other signaling molecules single neurons release. And I'm just gonna go over these. I can't resist showing you, however. So these are the channels, and we have their open channels here, and we have neurons in those single open channels. And you can see here that where is the neuron sending its processes? They're not down the middle, but they're along the edges. And I think this is a continuing theme. I bring this up because it's not something people have observed in two-dimensional cultures. But when you give the neurons an environment that's got more shape to it, they tend to seek surfaces. This is without any chemical cues. They like to have 2 sides touching. And this is again showing you in a 3-dimensional environment where the processes are going basically where 2 sides can touch the surface. And this is simply showing you that the neurons develop normally. They like to be in extracted PDMS. PDMS has platinum and other non-biocompatible substances in it, and therefore you enhance development, you enhance the outgrowth of processes. You get a more normal-looking neuron than if you simply use the stuff that comes off the shelf. If you want to know more about this, either ask Larry or look in this paper in Lab on a Chip. And this is more on survivability. This is showing you using these very low density cultures to use matrix crystals to extract peptides. And these are then the peptidomic peptides that are pulled out by mass spectrometry. And these are absolute identifications of these molecules, so we can discriminate two different types of cells co-cultured at very, very low densities based upon knowns, and then we can look at some of the other things they express. So the very, I got it, there's another slide. There was another slide here. Nevertheless, so basically, this is the last thing I wanted to say about biosensing, like the ultimate in biosensing and bioactuation. And that is to take everything that happens at the single cell level and put it back in the context of the organism. You know, we're really only studying the components, so we could put them back together and see how everything works. And so this is a study that was published in Nature Nanotechnology last month that Annika Jane, one of my students, kindly showed me. And what it involves is controlling the behavior of this very, very small roundworm, which is a very widely used bio model because it has every cell, every worm has a fixed number of cells and has a fixed number of neurons. You can identify every neuron, so you can really get at, you know, what does that neuron do for the way the animal behaves. But as far as neural basis of behavior, it's a very hot topic. And so what they did was they engineered nanoparticles that had basically a recorder. It was a part of a molecule that binds, like an antigen-antibody reaction, to a part of a molecule that was engineered into the worms. And so basically, these nanoparticles get put in the environment, they get taken up by openings in the worm, including this little mouth, and there they are in the environment of the sensory neurons that are what the worm is tasting its environment with. And these nanoparticles are sensitive to infrared heating. When they're in the membrane and they're in the proximity of one of the ion channels that, when it's open, lets calcium come into the cells, those neurons become active. So what they did with these worms, which are creeping, you know, in their little wormly way, crawling around, were to then turn on the radio frequency device and heat up the nanoparticles. And when the nanoparticles are heated up, they then open the calcium channel, and calcium goes in, activates the neurons, and inactivates the worm. And so, okay. I can't believe it won't show me the movie. I've done this so many times. Eek. Well, anyway. Best laid plans. I'm going to try this one more time. Strange things are happening here, I have to say. Well, be that as it may, we're going to get this technology down. I can't, it worked on my computer every time I did it. But nevertheless, what you need to do is go to Nature Nanotechnology and look at the movies. Because what it shows you is the little worms, and they're quite fetching really, they kind of wiggle around in their little environment. And normally, and then they turn on the radio frequency, nothing happens to the normal worm, unless it's been...
Martha, can we click them in and see what happens?
Goes on to my next slide. Well, anyway. But it's going blank on my computer too, that's what's weird, which it didn't normally do. Anyway, so when they turn the radio frequency device on, the worms sort of, their tails keep waving, but their front end stops. And then they turn it off, the worm starts to crawl again. Now, this of course is only a worm, but this is sort of the beginnings of ways in which you can think about engineering environments where you can control the activity of neurons, which in turn control behavior. And that's really the other part of my life. How does the brain control behavior? So basically, this brings me to the end of my talk. I hope that you'll come away from this with a feeling about, with a sense of the unusual structural and functional features of brain cells and the bases of biosensing and bioactivation of neurons, the importance of new technologies that involve materials, 3D environments, novel imaging. Oh yes, and last but not least, I promised I'd tell you something about questions that you could answer and discovery applications. Are these insights in neurobiology relevant to you? These are the types of neural-based diseases that are estimated by the National Institutes of Health with respect to total cases in the United States and the costs per year, hearing loss, all depressive disorders. Let's look for the really biggest one. I guess this is it, all depressive disorders. Then various genetic disorders, Alzheimer's, stroke, anything with the heart is big. And then various affective and motor disorders. Traumatic brain injury has emerged as a big new area. And then of course, injuries and multiple sclerosis. All of these are disorders based in the nervous system. And they're all disorders that are waiting for new kinds of therapies to address them. And that brings me to the end of my talk. This is just some of the things we talked about, and that the hope that these new technologies will let us see the brain in new ways. So thank you.